This video is Unit 3, Learning Targets 5 through 7, Cell Membrane and Membrane Transport. So a little bit about plant cell walls and just cell walls in general. Cell walls are structures that give plants, give cells protection, also give them shape and support. The reason that plants can grow to be as tall as they are is because of their cell walls. Fungi and bacteria also have cell walls. These are made of different materials than plant cell walls, but they do have cell walls nonetheless, and these cell walls function much the same as the cell walls in plants. Now, one thing I didn't mention, I will say again, I know I've said this before, plant cell walls are made of a carbohydrate called cellulose. Fungal cell walls are made of a carbohydrate called chitin. That's how you pronounce that. Now let's talk about the cell membrane for a little while. The cell membrane is composed of a phospholipid bilayer. Now, what do I mean by that? Well, you can see the phospholipids. We've talked some about them already. These are these little structures that look like heads with two legs on them. Phospholipids have two different sides to them. They have the hydrophilic head, hydrophilic meaning water loving. So this structure is also polar. It accepts water. And then there is this portion of the phospholipid, and it is hydrophobic, meaning that it is water fearing. And so what happens when you put these two these phospholipids in water is they will actually face one another, like you see there on the left. They will, the hydrophobic ends will face one another, and the hydrophilic parts will face out, because the hydrophilic heads have a phosphate there on the heads do, and this gives them a negative charge, and so they face out away, whereas the inside is these long fatty acid chains, and so they're extremely hydrophobic, and so they'll face one another. This gives the cell membrane a lot of protection. It allows the membrane to let certain things in and out, which is this concept of semi-permeability. The cell is semi-permeable, or the cell membrane is semi-permeable, meaning that some things are allowed in and some things are not. And some things are only allowed in at certain times. And so the cell has the ability to pick and choose what comes in and out. And that is in large part to the way the membrane is structured, with the hydrophobic inside and the hydrophilic outsides. Here's a picture of this of the cell membrane. It's a little more accurate as far as all the different structures that are involved. Sometimes the cell membrane is referred to as the fluid mosaic model, because this is the idea that all the elements of the cell, the proteins that you can see there, the proteins that are inside the cell membranes, this is just a giant protein that goes all the way through the cell. And there are some that just kind of sit there on the outside. There's some carbohydrates. These are steroids here, cholesterol actually. And all of these items exist together in fluidity. They aren't static, meaning they're not staying in the same place. They're continually changing places in order to facilitate the needs of the cell. See this, the proteins on the cell, those proteins are utilized to allow transport of materials in and out of the cell. The cholesterol there in the cell membrane actually gives the cell membrane flexibility. And so everything in the cell membrane has a purpose, and this whole concept is called the fluid mosaic model. So, talking about transport on the cell membrane, we've talked about solutions. Just to review, a solution is a mixture in which the solute is completely dissolved in the solvent. A recent test question that many of you didn't do well on suggested that what is it called when you take boiling water and you pour salt in it? Well, the salt will dissolve, causing there to be a saltwater solution, whereas the water is the solvent and the solute is the salt. So when we talk about concentration, this is the amount of solute per volume of solution. There's a simple equation that you could do with this. The mass of the solute divided by the volume of the solution. And so if you'll look at these two beakers, they're full of some sort of yellow dots, and they both have the same amount of yellow dots in the beaker. The difference is the one on the left is much more concentrated. Why is that? Because the one on the left has the same amount of solute but a much smaller volume, and so the concentration would go up. You're familiar with this concept. We've all drank Kool-Aid in our lives. Well, the Kool-Aid on the left is not concentrated at all. There's very little Kool-Aid for the amount of water. This is like Kool-Aid that's been sitting on the counter for too long with ice in it. Versus the really strong stuff on the right, it's like putting 2 Kool-Aid packets in 1 pitcher of water to make some particularly strong Kool-Aid. Very concentrated, lots more Kool-Aid per the amount of volume. Because notice all 5 of those cups have the same amount of volume, but they have different amounts of solute dissolved in them. There's the equation again. This is how you write it, 4 grams per liter. You probably won't have any of those types of problems, but I do want to introduce you to this concept because of what we're getting ready to talk about. So we're getting ready to talk about something called a concentration gradient. Now, a gradient, when you think of the word gradient, sometimes you've heard it used in like there's a certain gradient to this landscape. Well, maybe there's a hill on it and has a certain gradient. Or if you're familiar with that at all with the with roofing, well, this house has a a certain kind of grade to it, is what they would say. This house has a 3% grade or whatever. Well, a gradient in this case is very similar, except for there's no hill or there's no roof to go up. But there you can see that one side is definitely more than the other, right? Like the top of the roof is higher than the bottom of the roof. And so on the left side we have 9 different little stars, and on the right side we have 2. This is a concentration gradient. The left side is much more concentrated than the right side. And right over here in the middle, there's some sort of membrane, like a cell membrane. Well, concentration gradients allow for there to be movement of materials. This is how cells create the ability to do work. These materials will always move from an area of high concentration to an area of low concentration. Consider yourself at the top of the hill on your bicycle. You don't have to pedal down. You just have to let go, and your bicycle will move down the hill. So if this membrane in the middle allows the passage of orange stars, these orange stars will move across the membrane until everything is equal. And when everything is equal, we say that it is at equilibrium. This is a type of passive transport. Passive transport does not require energy because it's just going downhill. Now, what if we wanted to go back uphill? We wanted to go against the concentration gradient. If we wanted to move these 2 stars this way, well, if it doesn't require energy to go downhill this way, then it will require energy to go against the concentration gradient. And we'll talk more about that. So with passive transport, there are 2 kinds of passive transport. One of them is just simple diffusion. And diffusion is the movement of materials from an area of high concentration to an area of low concentration. It's just what we've been talking about. See at the top there, the there's a lot of concentration. There's a membrane that allows the passage of blue things, and over time they will move to where both sides are at equilibrium. And at equilibrium, there's an equal, so everything's moving this way. But at equilibrium, there's an even amount of movement both ways. There's just, everything's nice and simple, everything's at equilibrium, not a lot of movement, everything's just kind of staying still, but there's an equal amount of movement both ways, meaning that it's going to stay equal. So here's another picture of this. There's the solution there moving towards equilibrium. Here at the bottom, you can see what happens with two solutes. They move towards equilibrium. Now, cells use this movement from high to low in order to do work, in order to produce energy. Just like you would have lots of energy at the bottom of that hill if you rode down your bicycle, cells use this in order to create energy. Almost all of the energy in your body is created with a concentration gradient like this. Now, osmosis is another form of passive transport that is the diffusion of water across a semi-permeable membrane. The diffusion of water. So this picture shows you, I'm going to kind of draw my own picture. Very similar kind of picture here. Let's see. We have a beaker, and it's full of water, and in the middle is this membrane. Well, on one side of the membrane, I'm going to fill it with red dots. And let's just say that this membrane does not allow the passage of red dots because it is semi-permeable. So those red dots will not move. They will not go from high to low because the membrane does not allow them to move. But the water will move. So what's going to happen? The water, which side has a higher concentration of water? The right side has more water than the left side. And so the water will want to move from an area of high concentration to an area of low concentration. So what will actually take place is the water on the right will sink and the water on the left will get bigger until both sides are at an equal concentration. And this actually would probably just keep going until it was empty, consequently. Now, we have some terms when we're talking about this. Draw another picture. Same picture. Same membrane. The left side would be called hypertonic because there is more solute. Hypertonic. The left side, because there's not as much solute, is called hypotonic. Now, these words, hypertonic and hypotonic, are often a source of great confusion. Hypertonic and hypotonic. First of all, you can't just use them without a context. So you can't take this bowl of soup or whatever and this bowl of green soup and say that that bowl is hypertonic, because you're not comparing it to anything. These words are always used as a comparison. And also, the term hyper, think of the term hyper, what does it mean? Too much. And so, hypertonic has to do with the solute. It has to do with the red things in this particular instance. Hypertonic, more solute. Hypotonic, less solute. And water always moves from hypotonic to hypertonic. Hypo to hyper. Hypo to hyper. Water always moves from hypotonic to hypertonic, and it will stop moving when the cell is called isotonic. So it's very similar to equilibrium when both sides have an equal concentration of water. So this shows a couple of practical applications for this. There you have normal cells in an isotonic solution, equal number of solute on the inside and the outside. If you put a cell in a hypotonic solution, meaning if you were to take like a bacteria and just put it in fresh water, well, there's more solute inside the bacteria than there is outside, and so water will attempt to rush into the cell, and it will actually cause the cell to burst. With plant cells, they won't burst because of their cell wall until they become turgid. Well, same thing. If you put a cell in a very concentrated saltwater solution, the cells are inside the cell is less concentrated than outside the cell because the outside is hypertonic, inside hypotonic, and all the water inside the cell will go out, causing the cell to shrivel up. Have you ever poured salt on a slug before? This is what's happening. All the water inside the cell rushes out to make the salt be at equilibrium, and it can't do that. It kills the slug. So, the last type of passive transport is called facilitated diffusion. This is diffusion through the assistance of a protein channel. We will talk a lot about facilitated diffusion in here. Very simple concept. You can see here the red squares need the help of a protein channel, and they use that protein channel in order to get through the cell. Still high to low, but just going through a protein channel. So the concept of active transport, this is transport that requires energy. This is going against the concentration gradient from low to high. You can see that there it requires energy. It doesn't just happen on its own. Your bicycle will not get to the top of the hill just by you looking at it. You have to put energy into the system. Usually this means that the assistance of some sort of pump. It's kind of like hooking your bike up to a pump, an air pump, and just staring at the air pump. That won't actually air your tire up. You need to actually press down the pump in order to put air into the tire. So proton pumps, we'll talk about proton pumps, they work very similarly in that they're pumping protons across the gradient. It requires energy to do that. So a type of active transport is called endocytosis. Endo, inside, cyte, cell. And so this is when it takes really big particles into the cell. Two types of endocytosis. Phagocytosis, this is solid particles, and pinocytosis, liquid. So how does this work? Well, here's your cell. Here's a red bean or whatever. Not a bean. Beans aren't that small, but you get the idea. And this bean needs to get into the inside of the cell. Well, what the cell will actually do is it will like fold around the organism, or if it's a bacteria or the particle, whatever is coming into the cell, until that little particle has its own little vesicle to get into. Separate this stuff here. It's like an ear. Alright, and then this vesicle's inside the cell, and you can see the cell membrane will kind of fix itself, and it has a little piece of membrane around it, and the cell will digest it or whatever. That's called phagocytosis or pinocytosis if it's water. Now the opposite of that is exocytosis. And we talked about the endomembrane system, how the proteins leave the cell via exocytosis through a vesicle. There's a vesicle full of proteins or whatever. They're leaving the cell. The cell needs to get rid of waste or something like that. It just kind of pushes its way out of the cell via control Z. Just kind of pushes its way out of the cell, and it goes, it's the opposite of endocytosis.